Hello everyone, welcome to this short session on amyloidosis. Now amyloidosis is a rare condition which is characterized by abnormal deposition of misfolded proteins in various tissues and organs in the body. It is a very rare condition, and because it is a rare condition, it is misdiagnosed by physicians and medical practitioners because they do not come across cases of amyloidosis on routine basis. But the real fact is that around 50,000 people worldwide, they suffer from amyloidosis every year. In India, the cases of amyloidosis are on the higher end, and in Kerala in the last 1 decade has reported several types of amyloidosis like cutaneous amyloidosis, hepatic amyloidosis and gel-solin amyloidosis. So it becomes all the more important for us researchers who are working in the field of amyloidosis to make people aware, especially in Kerala, about amyloidosis. So in this small session, we would be covering about what is amyloid, what is amyloidosis, a brief history about amyloids, the properties of amyloids, pathogenesis, classification and the organs which are involved in amyloidogenesis. As we all know, all the activities in the cell, they are carried out by specific biomolecules called proteins. We all know that proteins are synthesized by the genetic information that is stored in the DNA. As and when required, the DNA shares this piece of information with the ribosomes, which are the protein synthesizing factories that are present in the cell. When these ribosomes, they receive this information in the form of RNA, they encode the proteins, and once the proteins are synthesized, they get folded and acquire a characteristic shape which is important for the functionality of the protein. Now, if by any chance the protein loses this folding trait or this fold, what happens is that the cell is equipped with a quality control machinery that can sense these misfolded proteins and remove it from the system. But under certain conditions such as inflammation as well as certain genetic disorders, what happens is that the proteins they would continuously misfold and they will not be cleared by the system because the quality control of the cell will fail under these circumstances. Once these proteins are misfolded, what happens is that the proteins they become more disobedient, and by disobedient what we mean is that they lose their ability to be degraded by the cells. So we all know that all the normal proteins in the body, they are biodegradable and recyclable. These amyloid proteins, they are not biodegradable and they are not recyclable. They stay within the cell and they create a lot of havoc within the cell, affecting the cellular activities, affecting the tissue functionality and resulting in organ damages. So amyloidosis can be regarded as a disease in which the abnormal proteins are resistant to be broken down, and these accumulate in the various tissues and organs and create a lot of organ dysfunction. If the amyloid builds up in the kidney, the heart, the liver, the gastrointestinal tract, the nervous system, it causes those organs or cells to function very poorly. Thus, the symptoms of amyloidosis are associated with abnormal functioning of the organs that are involved. So as you can see from the picture that is provided, the protein molecules in the newly formed cells, they undergo the folding process, and these folding is required for performing the biological activity of the cell. On the right hand side, you can see that the misfolded proteins, they are not removed by the quality control of the cell, and what happens is that they accumulate in various tissues. In this case, as the picture indicates, the misfolded proteins have accumulated in the blood vessel walls, and once they are accumulated and they accumulate over a long period of time, they can affect the permeability of the cell wall and resulting in various chronic conditions such as ischemia. So here in this case, the learner has to understand that there are 3 processes that are involved in amyloidosis. One is production, degradation and clearance. What happens during amyloidosis is that the production is on the higher end. Okay, so when the body or the tissue or the cell overexpresses that particular protein, what happens is that it becomes difficult for the cell to check whether the protein that is formed is of the correct conformation or not. So, this is one of the reasons why the proteins they misfold. Second reason is the degradation machinery, when they fail to clear or degrade or clear these amyloids from the system. That is also another reason for the protein buildup in the various tissues and organs. So, in this picture you can see how a folded protein has lost its conformation and has become misfolded now. Now, these misfolded proteins, they have a greater propensity to adhere, stick on top of another and form higher order structures called amyloid fibrils. Amyloid fibrils are very thin, long, unbranched higher order structures of diameter 17 to 13 nanometer. These are pathogenic in nature and they are formed by the process called polymerization. The mechanism of formation of polymerization varies in different amyloids because of the nature of the monomers that is present in it. The fibrillar proteins can also deposit either intracellularly or extracellularly. A very good example of this is the Alzheimer's disease. In Alzheimer's disease, you can see two separate types of deposits. One is the amyloid plaque deposit and the second one is the neurofibrillary tangle deposits. The amyloid plaque deposits are made up of amyloid beta monomers, whereas the neurofibrillary tangles are made up of tau proteins. The amyloid plaques are seen deposited outside the cell, whereas the neurofibrillary tangles are found inside the cell. Previously, it was thought that all the amyloid proteins are extracellular, they are deposited on outside of the cell, but that is not the case. A clear example of that is the neurofibrillary tangles, which are found intracellularly. It is found inside the neurons in the case of amyloid diseases. Let's take a break and come to the history of the amyloids. Now, the term amyloid has got its name from the Latin word which means amylum. In botany, this particular term is very familiar and it means starch-like. In medicine and research, the term amyloid was introduced by a great researcher called Rudolf Virchow in the year 1854. Rudolf Virchow was a great German pathologist who lived from 1804 to 1881. And he saw that the nervous system of certain diseased patients, they contained deposits which were starch-like. After many years, it was argued that these are not starch-like substances. Later, it was revealed that these are proteinaceous substances, but the term amyloid is still continued even today. Now, if you look into the chemical constitution of these amyloids, 95% of these amyloids are made up of fibrillary protein, and 5% of the amyloids, they comprise of P proteins, proteoglycans, glycosaminoglycans and serum amyloid P. How are fibrillary proteins formed? If we look into the formation of the fibrillar proteins, three distinct phases can be seen. The first one is the lag phase or the nucleation phase. The second phase is called the exponential phase or the log phase, and the third phase is called the stationary phase or the saturation phase. The nucleation or the initiation of the fibrils, they start off at the lag phase, and the buildup of the amyloid fibrils, they occur at the exponential phase. Now, if we plot a graph of the amount of fibrils that are formed versus the time, we get a characteristic sigmoid curve. A greater understanding can be obtained by looking at this particular model system, where researchers have proposed different types of models in order to describe the formation of the amyloids. If we look into the native like aggregation mechanism, what you can see is that the proteins, they unfold. They partially unfold, and these partially unfolded monomers, they come close to each other. They have this intrinsic or inherent tendency to adhere to one another, and they will adhere and then they will form a nuclei or a scaffold, which later builds and forms the amyloid fibrils. So, that is one way of amyloid fibril formation. In another model, that is the nucleated polymerization model, what is being proposed is that by a thermodynamically unfavorable mechanism, partially unfolded proteins or misfolded proteins, they come close together and they form a oligomeric state, and in this oligomeric state they later draw more monomers into that oligomeric state and later develop into an amyloid fiber. fibril. In the nucleated conversion model, what is stated is that these partially unfolded or the misfolded proteins, what happens to them is that they would rapidly come close together and they will form a disorganized amyloid oligomeric structure. And this oligomeric structure, it would reorganize itself and then they will form small nucleation points, which would increase to form larger nucleation points, and then from there the amyloid fibril will develop. That is one way. Another way is that once this disorganized oligomer is formed, it can attract more monomers into the oligomeric form, and then what happens is that it would reorganize itself and form a larger nucleation point from which a amyloid fibril will be developed. In a yet another model, what is proposed is that the mature amyloid fibril, that is the fibrils that have been formed, they would degrade or what you can say break down, and then these smaller fibrils, they will serve as nucleation points because we have so many N terminals, and these N terminals will attract more monomers and the amyloid would expand itself. During the degradation process, many nucleation points are also generated, and these nucleation points would again favor the amyloidogenic process. Now, can the amyloids be characterized? Yes, amyloids can be characterized by many techniques. An important technique is the physical techniques. What is being described over here is the transmission electron microscopy, the atomic force microscopy, circular dichroism, Fourier transform infrared spectroscopy and X-ray crystallography are all biophysical means to understand amyloidogenesis. What you see in the picture over here on the right hand side is the picture of mature amyloid fibrils that are taken by transmission electron microscopy. Here in this case, the amyloids, they are deposited onto carbon grids, and then they are stained by a negative stain like the uranyl acetate, and then viewed under the electron microscope, and we can see the picture of these amyloids. Here the mature amyloid fibrils, they would take up these needle like or long what you can say strand like structures. In the AFM, a smaller area only can be visualized, but the advantage is that we need not stain these amyloids. All we have to do is that we have to deposit these amyloids onto the mica and wash off the excess and view it under the microscope, and the structure of the amyloids can be understood by this particular process. But the drawback is that only a smaller area can be assessed by this particular means. CD and IR and crystallography, X-ray crystallography, it enables us to understand the extent of the secondary structures that are being formed in the amyloids. As you all know, the proteins, they have predominantly the alpha helical as well as the beta plated structures, whereas in the amyloidogenic proteins we can see that it acquires predominantly the beta plated structures, and these beta plated structures are signatures of amyloids, and the presence of these structures can also be biochemically evaluated by certain staining techniques as well. An important staining technique that is being used by histopathologists for amyloid confirmation is the Congo red staining. So, Congo red is an azo dye. What the Congo red dye does is that it will go and intercalate itself in the beta plated structure, and when these sections are stained by the Congo red, the amyloid stain would take up the deep pink or the red color. But when it is kept under polarized light, it would show the characteristic apple green birefringence. Thioflavin T, thioflavin S are other dyes which are used to study the amyloids. But even though there are a large number of staining techniques that are available, the most specific method to study the amyloid is still immunohistochemistry, because in this method we use specific antibody with which we can identify the type of amyloid that is in picture. Now, coming to the classification of amyloids. Amyloids can be classified as major amyloids and minor amyloids. The major amyloids, they include the AL protein, the AA protein and the A beta protein, whereas the minor amyloid forms, they include trans-thyretin, beta 2 microglobulin, serum amyloid P, proteoglycans and the sulfated glycosaminoglycans. Now coming to the major amyloid forms, we would be focusing here on 3 types, that is the AL type, the AA type and the A beta type. Now the AL protein is seen under certain cancerous conditions. Which type of cancerous condition, if you may ask? It is during this what you can say plasma cell cancer. In plasma cell cancer and in multiple myelomas, what we see is that we see plasma cell proliferation. There is a monoclonal proliferation of plasma cells. These plasma cells, they produce excess amount of immunoglobulins. Specifically, they would produce more of the light chain immunoglobulins. This light chain immunoglobulins would aggregate and they will form the AL deposits. Now, this AL deposits can affect multiple organs like the heart, the liver and the kidney. In the AA type of amyloidosis, which is predominantly seen during chronic inflammatory conditions, specifically when the liver is affected, when there is chronic inflammation that is taking place in the liver, there is a lot of pro-inflammatory mediator production. Specifically, the interleukin 6 and the interleukin 1 levels are very high, and this overwhelming response will induce the production of acute phase proteins like the serum amyloid protein, and excess amount of serum amyloid protein would misfold and result in the formation of AA deposits. The A beta protein is normally seen during the Alzheimer's disease. Alzheimer's disease is a progressive neurodegenerative disorder, and in Alzheimer's disease predominantly the amyloid plaques are seen, and the amyloid plaques are seen deposited in certain regions of the brain, specifically in the cerebral cortex and the hippocampus. The monomer that we see in the amyloid plaque is A beta. A beta exists in different forms. Lengthwise, from it can vary from around 39 amino acids to around 43 amino acids, and the predominant form that we see over there is the A beta 42, which is 42 amino acids in length, and this particular A beta 42 peptide has got a greater propensity to aggregate, and they would aggregate and form the amyloid plaques. In this picture, you can see how the plasma cell proliferation occurs and how the antibodies, they would misfold and form the amyloids, which would deposit in the gastrointestinal tract, it can deposit in the heart, and it can even deposit in the liver. Now, we will look into a clinical case that has been reported very recently. This is the autopsy finding of a patient with primary systemic AL amyloidosis, where you can see liver enlargement, you can see heart enlargement, and you can also see kidney damage, extensive kidney damage that is occurring. Here the authors of this case report, they have reported that extensive amounts of amyloids were seen in the myocardium, and this has led to the thickening of the valves and the chambers of the heart. And in the liver, excessive amount of this amyloid deposition, what it has done is that it has induced a lot of what you can say pressure in the portal veins, and that extensive pressure, which is normally seen in conditions such as cirrhosis, was evident in this amyloidosis condition. The kidneys were also extensively damaged, as evidenced by asymptomatic proteinuria. There was a lot of damage that the proteins escaped into the urine. But the authors, they also state that despite the coexistence of a rich clinical feature and availability of a diagnostic method, this particular case of amyloidosis was confirmed only after the death of the patient, meaning the patient had multiple organ failure, the patient had different types of issues like unexplained weight loss, liver failure, progressive renal insufficiency, yet this disorder was not identified. It was only after what you can say the death of the patient, autopsy had revealed that all these organs had extensive amyloids in it. So, the authors also state that vascular lesions in the skin followed by multiple organ failure need to be considered by physicians in order to evaluate whether the condition is amyloidosis or not. So far we have discussed about three aspects. One is overproduction of the amyloid, and second one is there is production but then there is no degradation and clearance. Now we also talk about another particular condition where there is normal production of the protein but it is in the mutant form. An important example can be cited by the case of trans-thyretin amyloidosis. So, in trans-thyretin amyloidosis, the protein that is overexpressed is trans-thyretin. Trans-thyretin has got physiological functions in the body. It is a transport protein that helps in the transport of thyroxine, which is a thyroid hormone, and it also helps in the transport of retinol, which is vitamin A. Now this trans-thyretin or TTR, it is comprised of 4 subunits. Under certain conditions, what happens is that these 4 units they can't be held together, they become highly unstable, they separate out, and then they would aggregate in a different form and form the amyloid fibril, which we refer to as the TTR fibril in the case of trans-thyretin amyloidosis. Now there are 2 different types of amyloidosis. In the wild type TTR condition, the normal protein becomes unstable and it will break down and it will form the fibrils, and this particular condition is seen in the heart of aged patients, and the condition is referred to as senile cardiac amyloidosis. In the mutant form of TTR, what we see is that the liver produces the TTR, but the TTR would destabilize and then it would form amyloid fibrils and it would deposit in the heart and the nerves and result in a condition called familial amyloid neuropathy, where either the heart is affected or the nerve is affected or both the what you can say tissues are affected. The Cleveland Clinic has set out this picture as part of their awareness campaign, where they indicate that numbness, burning or tingling of the arms and legs, spinal stenosis of the back, lightheadedness upon standing, shortness of breath, palpitation, chest pain, fatigue, poor appetite, blotting or excessive gas production, diarrhea and constipation. They can all be signs that the patient is suffering from TTR amyloidosis. How can amyloid buildup occur in the tissues? So, previously I mentioned that the cell has got a quality control mechanism through which this misfolded proteins can be removed from the system. The first one is the proteasomal system, where the proteins which have become misfolded, they can be ubiquitin labeled and then carried over to the 26S proteasome, and they can be degraded by the 26S proteasomal machinery. That is one way. The second way is that if the deposits are found outside the cell, then the system can recruit professional phagocytes like macrophage cells, which can come to the site and then what you can say eat up these misfolded proteins. So these are the 2 quality control mechanism that is adopted by the body in order to clear the misfolded proteins. Now certain conditions where the clearance or the degradation becomes difficult, that is when the amyloid deposition occur. As in the case of AL protein amyloidosis, which we have previously mentioned that it is a case of plasma cell proliferation, there is increased amount of immunoglobulin, specifically the light chain, and the abnormal folding of these light chains, and in addition to that reduced proteolysis can result in the deposition of the AL protein. In yet another case, that is the AA amyloidosis, we talked about chronic inflammation of the liver. There is macrophage activation, overwhelming production of pro-inflammatory cytokines like interleukin 1 and interleukin 6, which elicits acute phase protein production, specifically here in this case the serum acute phase protein, and they can actually lead to the deposition of the AA type of amyloid protein. In the ATTR amyloidosis, we have seen that mutation in the TTR gene, specifically the point mutations, what it does is that it will result in the normal amount of protein. The protein levels are normal, but whatever protein is being deposited are mutant forms. They cannot do their function, they aggregate and they deposit in the organs and resulting in organ failure. That particular disease type is called the ATTR type of amyloidosis. Now let us look into the mechanism of damage caused by these amyloid fibrils. Amyloid fibrils can induce mechanical stress. Say for example, the amyloid deposits that are found in the brain of neurodegenerative diseases like Alzheimer disease. They cause a lot of mechanical stress and they will affect cellular function, resulting in what you can say organ damage. In the case of blood vessels, what happens is that the amyloids, they accumulate over a period of time. It can affect the permeability of the cell wall, resulting in ischemia. That is one way of how the amyloid fibers can affect cells and tissues. There is another case of direct cytotoxicity. In the case of direct cytotoxicity, say for example in AL amyloidosis, what we see is that there is an increased production of the immunoglobulins, specifically the light chain immunoglobulins. These light chain immunoglobulins, which are produced by the plasma cells, they create toxicity for the heart cells. So in this way, a protein that is produced by one type of cell can affect another type of cell. Then there is the what you can say pre-fibrillar toxicity. Pre-fibrillar toxicity is again in the case of amyloids. The mature amyloid fibrils may not be toxic to certain types of cells. Once they are released from that particular system where they are produced, say for example the brain, they can escape because owing to compromise in the blood-brain barrier, the amyloids which are produced by the brains can cross the barrier and they can reach other organs as well. Under these conditions, the amyloid fibrils, they do not pose much toxicity. But the toxicity is imparted by the pre-fibrillar forms, that is the species which are formed prior to the amyloids, mature amyloids, those pre-fibrillar forms, or the oligomeric forms, that is the species, the intermediate species. These are the ones they pose toxicity to the cells. In Alzheimer diseases, the A beta oligomers have been found to be neurotoxic. Classification of amyloids. Now, amyloids can be or amyloidosis can be classified into three types: general or systemic, localized and hereditary. Primary amyloidosis, that is conditions where we saw AL protein being accumulated and conditions where we saw the AA protein getting accumulated. These are all cases of general amyloidosis. Then there is a third type called the hemodialysis associated. In the hemodialysis associated amyloidosis, what we see is that patients who undergo dialysis for a prolonged period of time, they are predisposed to this particular type of amyloidosis. However, in today's time, the dialysis is more efficient. All the proteins are cleared from the system. Previously, it was observed that certain proteins like the beta 2 microglobulin, they would escape dialysis, they accumulate in the kidney and then they would induce amyloidosis in that particular organ. So, all these three cases, that is the primary amyloidosis that involves the AL protein, the secondary amyloidosis involving the AA protein and the hemodialysis, they all come under the general category of amyloidosis. In the localized amyloidosis, we can cite two specific examples. One is the senile cerebral type and the endocrine type. The senile cerebral type is what we have previously discussed, is about the Alzheimer disease, where the amyloidosis occurs only in specific organs. In Alzheimer disease, the amyloidosis is seen localized in the brain, that too in specific regions of the brain like the cerebral cortex and the hippocampus. So, here what happens is that there is a transmembrane protein called the amyloid precursor protein. The amyloid precursor protein gets cleaved by various secretases, resulting in the formation of amyloid beta. Amyloid beta 42 has an increased propensity to aggregate and they would form the amyloid plaques. In the endocrine type, two cases can be cited. One is the carcinoma of the thyroid and the second one is type 2 diabetic mellitus. In the carcinoma of the thyroid, it has been observed that a peptide called calcitonin, which has got hormone functions. These peptides, otherwise they have an increased propensity to aggregate. These peptides, they are overexpressed during the carcinoma conditions and they would aggregate and get deposited in the thyroid gland. That would result in amyloidosis in that particular location. In type 2 diabetes, it has been observed that islet cells, they produce a particular peptide called the amyloid peptide, islet amyloid peptide, and this islet amyloid peptide gets deposited in the islet of Langerhans and resulting in the formation of amyloids in the islets. Hereditary types, it has been classified into 3. The first one is the familial Mediterranean fever, the second and the third is familial amyloidotic neuropathy and systemic senile amyloidosis. In familial amyloidotic neuropathy and in systemic senile amyloidosis, what is seen to be aggregated is the TTR forms. So, in both these cases, it has been observed that trans-thyretin or TTR, the tetrameric form, it would degrade and then it would reorganize itself and form the fibrils. In the familial Mediterranean fever case, again the acute phase protein is seen to be overexpressed and misfolded, and this normally is seen under conditions of liver inflammation. Now, there is the secondary amyloidosis cases. Secondary amyloidosis cases occurs when there is already an underlying chronic condition. Say for example, people with tuberculosis, people with osteomyelitis, people with rheumatoid arthritis, spondylitis, inflammatory bowel diseases, they all have this case of secondary amyloidosis where you can see amyloids getting deposited in their organs. So, these patients who suffer from this syndrome, they also have a greater chance of developing amyloidosis. Drug abusers who repeatedly inject drugs into their skin, they develop lesions, and over there also in specific areas this amyloidosis can occur. Amyloidosis can also occur under certain solid tumors like the Hodgkin's lymphoma condition. Now, let us actually look into the organs which are being affected by amyloidosis. In the case of primary amyloidosis, the gastrointestinal tract, the respiratory tract, peripheral nerves, skin and tongue are primarily affected in primary amyloidosis. In secondary amyloidosis, we see endocrine glands coming in apart from kidney, liver, spleen, lymph nodes, adrenal glands and thyroids are also largely affected with during secondary amyloidosis. In familiar Mediterranean fever, what is seen is the kidneys, the blood vessels, spleen, respiratory tract and the liver are affected by amyloidosis. Okay, having said that amyloidosis is a rare condition, it need not be forgotten that amyloidosis cases can occur in anyone, and there are treatment centers that are there in India which can effectively treat amyloidosis. The BLK Super Speciality Hospital in New Delhi, the Apollo Hospital in Chennai, the Apollo Glen Eagles Hospital in Kolkata, the Medanta Hospital in Gurgaon, in the Max Speciality Hospital in Saket, New Delhi, Fortis in Navi Mumbai and Columbia in Bengaluru. They are hospitals and centers that treat amyloidosis. Thank you for listening to this short session on amyloidosis.